Welcome back to Synergistic Science. I'm Dr. Goelzer, and today we're going to be discussing the topic of differential gene expression, or the idea that is commonly seen in developmental biology of a multicellular organism having cells with different fates or being differentiated. Big picture, the idea is the human body has multiple cell types, things like skin cells, reproductive cells, bone cells, muscle cells, and all of them should have the same genome, but are expressed differently, and you would expect to see a different transcriptome and a different proteome. This idea is known as genomic equivalency, or the scientific theory that every cell of a multicellular organism, such as a human or some of our developmental model organisms like zebrafish or mice, should have the same genome, the same DNA code in every single cell of that organism. Differential gene expression is the big concept that although every cell in a multicellular organism contains the same genome, the specific proteins that are expressed in different cell types will be wildly diverse. So every somatic cell or body cell, non-reproductive cell nucleus of an organism contains a complete genome, or the whole DNA code that is established in the fertilized egg. The DNA of all differentiated cells is identical. So if you've ever seen the sequencing kits, things like 23andMe, where you spit in a cup and then you get to see your whole genome, it doesn't matter what cell we take the DNA from. So the cheek cells that may have sloughed off into my spit should be sequenced the same as if I took a skin cell from another part of my body or took a more invasive sample of, I don't know, bone or muscle. The unused genes of differentiated cells are not going to be destroyed. They're not thrown away. It's not like a skin cell goes, oops, I don't need those muscle cell genes, basically. It keeps all of them, but and retains its potential for being expressed, even if it's not actually using it. Only a small percentage of the genome is expressed in each cell. It depends on each cell type. So a certain group of genes would be expressed in your bone cells, and a different group of cells would be expressed in your liver cells. A portion of RNA is synthesized in each cell, and it's very specific for each cell type. So let's look at the process of how we actually get this differential gene expression. There are 6 main steps that will be gone through within a given cell to actually get from the DNA code to actually expressing a final product. Okay, so you can see on the left here, we start with our double-stranded DNA, and it will go through the process known as the central dogma, going from DNA, big picture, to our single-stranded mRNA code, and then that mRNA gets made into protein. So the central dogma is the transfer of information from DNA to make proteins. DNA is transcribed into RNA. So we're transcribing here in the nucleus, making a messenger RNA or an mRNA that will code for protein. It can leave the nucleus, and then it can get translated, or it can go through the process of making codons of mRNA and translating it into an amino acid sequence or a polypeptide chain. If we look a little more deeply into the image on the left, you can see the double-stranded DNA getting made into the pre-mRNA through a process known as transcription. And then that pre-mRNA goes through a processing process, I guess, here, where it would be capped. So you have a 5' cap here, a poly A tail, and you'll also notice the color differences showing us that it has been spliced, or only looking at the exons and introns, basically, of that sequence. The exons being the ones that are expressed or kept. Then once it has been processed, it can be transported out of the nucleus as a mature mRNA. And then we come to step 4, where that mRNA finds a ribosome and the codons can be read and turned into an amino acid chain through the process known as translation. Then this sequence of amino acids, which would be kind of a primary structure of the protein, can then be folded properly, thinking kind of primary, secondary, tertiary folding. It can be modified, so if it needs any phosphorylation or sumylation, any special chemical groups added to it, and then it can finally carry out its function. There are 4 main levels from this big picture process where we can regulate the gene expression. Effectively, they're like dials of sorts that we can adjust to get a wide range of different cell types. So our first level is differential gene transcription regulates which of the nuclear genes are transcribed into pre-mRNA. So at that first level, this is where we decide which sequences of DNA are actually going to get made into pre-mRNA. And this is regulated largely by how accessible genes are. So this would be things like nucleosomes or histones. How tightly is it wound? Is it actually accessible? And then also by any transcription regulators or transcription factors. So are we going to have any proteins coming in, binding, and actually making the RNA? The second level is selective pre-mRNA processing, regulates which parts of the transcribed RNAs can enter the cytoplasm and become mRNAs. So the processing steps from going from pre-mRNA to a mature mRNA would include capping, tailing, and splicing. So at this step, if it does not get capped and does not get tailed, it will very likely not end up out in the cytoplasm and it can't find a ribosome. So that has to occur. We're going to decide which ones actually can leave. And then also this step encompasses any sort of splicing. So sometimes there's alternative splicing where some exons are kept, some aren't, and it depends on cell type. Level 3 is selective mRNA translation, regulates which of the mRNAs in the cytoplasm are translated into proteins. Generally speaking, the longer the mRNA stays in the cytoplasm, the more likely it is that it is going to get translated into protein, and the more likely you are to get many protein products. The timing of that is regulated by the length of the poly-A tail. So the more A's that you add on to the end of the mature mRNA, the longer its half-life is going to be. Additionally, there's been some new papers coming out showing that ribosomes actually have preferences towards certain mRNAs. So while we don't fully understand that process, there is some choice effectively in which mRNAs will be translated and how often. And then the final level, level 4, differential post-translational protein modifications regulate which proteins are allowed to remain and which ones will function in the cell. So this is the step where you can add an acetyl group, a methyl group, phosphorylate a protein. You could label it for degradation if it needs to be removed, or maybe you need one of those special groups to actually help it function or perform a particular process within the cell. So this takes us through the basic steps of differential gene expression and how we can modify them within a given cell.